One other important factor which makes CRISPR a more desirable gene editing tool compared to others is the fact that it can simultaneously target many sequences at once by modifying the protospacer regions with different sequences. When the CRISPR locus is then transcribed, it will be degraded into fragments consisting of the CRISPR RNA and each of the different protospacer sequences. These will then associate with the Cas9 enzyme and be directed to each of the target sites complementary to the protospacers. CRISPR therefore offers the advantage of multiplex genome engineering, targeting multiple sequences at once. This is also inexpensive because all it requires is the modification of the protospacer regions. Theoretically, this can be achieved using other gene editing technologies, targeting multiple DNA sequences. However, they require much more expensive protein engineering constructive steps to produce their DNA binding domains. Even so, using CRISPR to target one DNA sequence is inexpensive, since it requires the simple modification of the protospacer region rather than the expensive construction of DNA binding domains. So CRISPR offers many advantages. First of all, it is inexpensive and you're able to target multiple DNA sequences at once. As well as this, it has very high efficiency in terms of targeting the DNA sequence of interest, making it a more desirable and better gene editing tool. One problem that does still remain is the induction of off-target activity with using CRISPR, which raises safety concerns due to the unanticipated effects of unwanted DNA binding. When targeting genes with gene editing technology, it is important to note that the DNA sequences between genes will differ greatly. The number of applicable target sites to be recognized by each technology will therefore differ. We also need to carefully select target sites which won't be similar to any other DNA sequences to which the technologies may also bind to. Therefore, each gene chosen to be targeted by the gene editing tools will be subjected to a different extent of off-target activity, making it difficult to compare the off-target activity of using the gene editing tools. Although CRISPR offers its advantages, such as the ability to multiplex, its inexpensiveness, and its high efficiency, which makes it the most desirable gene editing tool. One problem which remains, however, is its induction of off-target activity. One way to reduce the off-target activity of CRISPR is by increasing the specificity it has to its target site. One way in which this can be achieved is by mutating the Cas9 enzyme so that it induces single-stranded breaks within the DNA, otherwise called nickase activity. Similar to zinc-finger nucleases and tallins, we can direct these enzymes to sequences which are adjacent to each other on opposing DNA strands. Each of the enzymes in this case, directed by protospacers matching adjacent sequences, will induce a single-stranded DNA break, which overall creates a double-stranded break in the DNA. Another similar approach involves mutating and inactivating the Cas9 enzyme, then conjoining this enzyme with a Foc-L endonuclease. Cas9 will associate with CRISPR RNA and protospacer sequences. By generating two protospacer sequences which are able to match DNA sequences adjacent to each other on opposite DNA strands, we are then able to induce a DNA double-stranded break by the induction of two single-stranded breaks from each Foc-L endonuclease.